Once scientists could finally see microbes, a deeper challenge emerged: how to separate one from another. Under the microscope, every sample was a crowd, a swirl of shapes, each dividing and competing, impossible to distinguish in their natural chaos. To understand microbes, science had to learn isolation, not just observation. That breakthrough began in the late 19th century. In his modest German laboratory, Robert Koch sought to identify the cause of anthrax. He noticed that when diseased cattle blood was placed on a glass slide, tiny rods multiplied. But which one caused the disease? Koch realized he needed to study a single strain, free from all others, a pure culture. Only then could cause and effect be proven. His method was both simple and revolutionary. Koch spread bacterial samples across solid surfaces made from potato slices and later gelatin. Each bacterium multiplied into a separate colony visible to the naked eye. A microcosm born from a single cell. From these colonies he could isolate pathogens, inject them into healthy animals, and reproduce disease with precision. The logic of infection was no longer mystery but experiment. From this came Koch's postulates, a framework that turned microbiology into an exact science. The idea of the pure culture did more than identify pathogens. It redefined life itself. It revealed that every microbe has its own needs, rhythms, and metabolism. When Louis Pasteur and Sergei Winogradsky began cultivating microbes under controlled conditions, they discovered that bacteria were not just destroyers, but chemists of the planet. Some consumed oxygen, others thrived without it. Some produced acids from sugar, others drew energy from iron or sulfur. The study of microbial metabolism, physiology, was born. By the early 1900s, new tools transformed this discipline. Martinus Beijerinck introduced the enrichment culture, a technique that encouraged specific microbes to flourish by tailoring nutrients and conditions. Through it, he isolated nitrogen-fixing rhizobium and the tobacco mosaic virus, the first virus ever identified. Microbiology was no longer limited to disease. It became the study of life's hidden engines. The lab bench became a model of ecosystems in miniature. In these early cultures, scientists began to see patterns that still define biology today. Bacteria grew exponentially, then plateaued as nutrients dwindled. Competition and cooperation appeared even in glass flasks, one species producing byproducts that another consumed. Microbes revealed the universal logic of survival, adaptation through metabolism. To watch them grow was to witness evolution in fast forward. As techniques advanced, microbiology became a discipline of both control and curiosity. Sterile glassware, pure media, and precise incubation created repeatable experiments. Yet every discovery pointed back to nature's complexity. Even in isolation, microbes hinted at the vast ecological networks from which they came. Communities exchanging molecules, shaping soil, ocean, and atmosphere. The pure culture was never truly pure. It was a simplified echo of life's web. By mid-century, microbial physiology laid the foundation for molecular biology. Understanding respiration, fermentation, and biosynthesis opened paths to genetics, antibiotics, and biotechnology. From Koch's glass plates to modern fermenters and bioreactors, the principles remain the same. Control, measure, observe, and respect the vitality of even the smallest forms. The discovery of the pure culture did more than isolate microbes. It united science. It bridged medicine, chemistry, and ecology, showing that the same laws govern disease, decay, and renewal. It turned the microscope into a tool, not just for seeing, but for understanding. And it reminded humanity that life, in all its diversity, can begin again from a single cell.